In this lesson, then, we'll examine more closely what happens at the synapse, that junction between two neurons. Now here's a picture of two neurons. Here's a neuron, the cell body here, and the cell body here, two neurons, and here's the axon from one of the neurons ending in 3 terminals, so making 3 synapses on the target neuron. One of the synapses is on the cell body, and these 2 synapses are on dendrites. Now if you magnify that synapse, it looks like this. Notice the terminal does not physically make contact with the target cell. There's a space. So the axon terminal is packed with membrane-bound chambers called vesicles. And vesicles contain neurotransmitters. These are chemicals that can electrically excite a local region of a dendrite of the target cell. This is an electron microscope picture, so a very thin slice and high magnification scene of the terminal. Here's the axon terminal here, making contact, making a synapse with this dendrite here and as well over here, this branch of the dendrite. So inside the terminal we see these tiny little sacs. They're called vesicles. In fact, we see these other larger potato looking like things. These would be mitochondria. They generate energy for the cell. But we're going to focus on these tiny little circular shapes here. These are called vesicles. It's inside these vesicles that neurotransmitters, the chemicals involved in synaptic communication, are located. So what the terminal is going to do, it's going to be releasing the chemicals that are stored in these vesicles, and those chemicals are then going to influence, electrically excite the target cell, the dendrites here. So vesicles must fuse with or join the axon terminal membrane in order to release the neurotransmitters into the synaptic space. In the electron microscope picture, some vesicles are caught in the act of fusing. So over here on the left, this is our cartoon. Here again we have the synaptic terminal. We've got these chambers, these vesicles, and then the little green things would be the chemicals, the neurotransmitters. So what needs to happen is that these vesicles have to fuse with the membrane of the terminal here, so that the contents of the vesicles, the chemicals, the neurotransmitters, can be dumped out into the synaptic space. In fact, here we see again an electron microscope picture, a thin slice of a synapse, and we see the axon terminal here. This top part is the axon terminal, and then the bottom region here is the dendrite of the target cell. And we can see vesicles in the axon terminal, and in fact, circled in red here we've caught one of the vesicles in the act of fusing with the terminal membrane. What we can't see are the chemicals in there. The neurotransmitters are too small to see with the electron microscope. So we've got another fusing vesicle right here and another one here, and then these vesicles in here have not fused. So now let's go through the process step by step. We have an electrical signal arriving at the terminal, and in the next lesson we'll figure out how that electrical signal travels down the axon to the terminal. But the electrical signal gets to the terminal, and the effect of that is that these vesicles move towards the terminal membrane and fuse with the membrane, dumping their contents, the neurotransmitters, into the synaptic space. So the arrival of the action potential causes the release of chemicals from the axon terminal, and the chemicals diffuse across the synapse and bind to protein receptors on the target cell. These chemicals don't know where they're supposed to go. They're just bumping around here, but it's such a small distance here. It doesn't take long for them to bump into the target cell. On the target cell are proteins that have little binding sites for these chemicals. Now right now this is shown to be closed. It's kind of a channel, and if the neurotransmitter links up to the protein receptor, this channel will open up and allow the movement of charged particles like sodium ions into the cell. And remember, whenever we have a movement of a charged particle somewhere, it's an electrical current. So we've got neurotransmitters are being released from the axon terminal. They move across the synaptic space, and they're going to bump into the target cell membrane. But on the membrane are lots of channels, these receptors, that have a binding space for that chemical. And if the neurotransmitter binds to the receptor, the channel opens up, allowing the movement of charged particles into the cell. And that'll look like this. So here we have the neurotransmitter has connected to the receptor. The channel opens up, and this allows sodium ions to rush in. So this again is how neurons are electrically excitable. They have these channels in their membrane that permit the movement of charged particles, in this case into the cell. So this would be an electrical signal being generated as a result of activity at a synapse. And this is what we mean by communication, that the communicating neuron is activating the target cell by releasing chemicals that generate a small electrical signal in the target cell. Now, neurotransmitters diffuse away or are absorbed back into the axon terminal, and this is important because it stops the communication then between the two neurons. The synapse must be made ready to respond to another action potential. So if these neurotransmitters were hanging around for a long period of time, they would keep on opening up the channels, and then this synapse could not respond to the next action potential that came down to the terminal. So what happens is that these neurotransmitters just diffuse away and are taken up by other nearby cells, or are imported back into the terminal to be recycled. But this has the effect of stopping the communication so that another round of communication can happen in the future.